Hello and welcome to all my dear students. So far we have started with the next chapter, that is Genetics Part 1, Principles of Inheritance and Variations. Yes or no? In our last class we have also discussed some important terminologies, plus we saw the work of Mendel in the form of monohybrid cross. In my last class beta, I have explained you that how Mendel performed monohybrid cross and what was the result of monohybrid cross. Today, we are going to continue from there and we'll see that on the basis of monohybrid cross, what Mendel proposed, what was the outcome, what Mendel could understand with the help of the results of monohybrid cross. So today, I'll be continuing with monohybrid cross and we'll see different laws proposed by Mendel on the basis of monohybrid cross. Correct? Okay, so what is monohybrid cross? Just do a quick revision. It is basically a cross which is made by studying a pair of contrasting trait or one contrasting character. Correct? Now, for example, you know Mendel chose 7 genes controlling 7 different characters. So he could prepare 7 monohybrid cross. For each character, 1 monohybrid cross. So the 7 characters chosen by Mendel were like stem height, that means plant height, pod color, pod shape, seed color, seed shape, flower position and flower color. So these were the 7 characters chosen by Mendel, and one of the character which I chose for my monohybrid cross was T gene. That means T gene that is responsible for controlling height of the plant. Now beta, to start the monohybrid cross, we know that the first step is cross-pollination. Before cross-pollination, what has to be done? Selection of the parent has to be done. And the parents are always taken pure line. Pure line homozygous dominant for a character and pure line homozygous recessive for a character. That means if we have to start the cross, first step is what? This all I told in my last class, but still let's do a quick revision. Selection of parent. And how are the parents? Parents are always pure line. Correct? Pure line means homozygous dominant for the character and homozygous recessive for the character. Since I chose T gene, that means stem height, to study monohybrid cross, that is why I am using this TT and small t. You know the method of gamete formation, that also I taught. So suppose these are my parents, you know one we have to consider male and one we have to consider female. Male means whose pollen grains are going to be used, and female means whose female gamete egg is going to be used for fertilization. So first step is done. Second step is beta performing cross-pollination. So before cross-pollination, gametes have to be formed. Now here you see beta, both the parents are homozygous for the given character. So here the degree of heterozygosity, value of n will become 0. So anything raised to power 0 is equivalent to 1. Hence here the gametes produced from both the parents will be of one type. This male is going to produce capital T gamete and female is going to produce gamete having recessive allele of the T gene. Now what happened beta? These gametes, they are made to fertilize to form zygote, and then zygote is made to develop into new organisms. So what Mendel observed? That all the offsprings of F1 generation were tall. Now remember beta, tallness is the phenotypic behavior, whereas this form of tallness is the genotypic arrangement. So Mendel beta could not see the genotype. See, to understand the genotype, you have to look inside that what is the gene arrangement on the chromosome. But to observe the phenotype, Externally you can see. So Mendel was actually doing external study. He was just able to observe the phenotype of F1 generation, and to his surprise, he found that all the offsprings are coming tall. None of them are coming dwarf. He got surprised that, Arey Baba, one of my parent was tall, another was dwarf. So is that female is not giving anything? Why the female character is not coming in any of the offspring? To know this, he then performed the third step, which is selfing of offsprings. Now, as he chose beta pea plant, so you know pea plant is already bisexual, shows bud pollination. So selfing was a very easy step for Mendel. He had to do nothing, just left the F1 generation offsprings to perform self-pollination by themselves. So what will happen now, beta? When selfing will take place, now you have to actually cross the same genotypic parent with the same genotypic male and female parent. So this is the genotypic constitution of male. This is the same as female. So here there will be two types of gametes. As there is degree of heterozygosity, T is present in its heterozygous state. These are the two types of alleles. And here also these are the two types of gametes, even having one dominant allele and another recessive allele. So what he found? Then beta, he obtained the Punnett square. Beta, see, this all we have done in our last class. It's just that we are having a quick revision so that I can continue with today's class. Right? So this is how the Punnett square was. Now where he saw that the phenotypic ratio of F2 generation came as 3:1. That means 75% appeared to be tall and 25% appeared to be dwarf. So now Mendel was satisfied that at least at the end, after completing cross-pollination and self-pollination, Mendel saw that at least 25% of the offspring were coming dwarf, were carrying the initial phenotypic arrangement of the parent. Right? This much we have discussed in our last class. Now beta, as I already told you, Mendel was just able to observe the phenotypic ratios. He was just able to see the morphological appearance. Like here in F1 generation, he could only see that all organisms are coming tall. However, he did not know the genotype behind it, that whether these tall organisms are due to homozygous dominant condition or heterozygous. Similarly, here also, when he tried to obtain the phenotypic ratio of F2 generation, that was clear that 75% of the offsprings are coming tall and 25% are coming dwarf. Now, dwarf genotype is well understood. There is only one condition, homozygous recessive. However, Mendel could not understand once again that if 75% of the offsprings are coming tall, then whether are they homozygous dominant or they are heterozygous dominant. So, in order to calculate the genotypic ratio of both F1 and F2 generation, Mendel performed test cross. Phenotypic ratio, I hope, is clear. What is the phenotypic ratio of F1 generation? Phenotypic ratio for F1 generation is clear. 1. Why 1? Because all offsprings are tall. Then phenotypic ratio of F2 generation 3:1. Here all tall. Here 75% tall and 25% dwarf. Now he wanted to know the to know the genotype of both F1 and F2 generation, Mendel beta performed test cross. Now, what is this test cross? Correct? Test cross beta simply means crossing the unknown organism whose genotype has to be known with one of the parental type which is having homozygous recessive combination for the given gene. So what is test cross? Right. Test cross means performing the cross between unknown genotype organism, that means the whose genotype we have to calculate, with homozygous recessive parental type, with homozygous recessive parental type. So here the gene for the given character. So here the given character is height controlled by T gene. So what will be the combination of the parental homozygous recessive? It will be, right? So now Mendel performed test cross. Chalo. So F2 generation phenotypic ratio PR is 3:1, right? Where 3 means 75% are tall. Now beta, these tall may be either due to homozygous dominant or they may be due to heterozygous dominant. Correct? Now he performed test cross for all 75% of the offspring. All 75% of the offspring went under test cross. That means they were made to fertilize with homozygous recessive parental type. Now what will happen? In the first condition, in this condition, all offsprings will appear tall. Why? Because it is homozygous dominant and homozygous recessive. Then one of the gamete from homozygous dominant, one of the gamete from homozygous recessive, they will fertilize and all offsprings will appear tall with this genotype, heterozygous dominant tall. Now, if this is the genotype of the unknown offspring, then what will happen? See. So this is going to give gamete having recessive T, and this unknown genotype will give two gametes, one having dominant T, one having recessive T. Now when they are made to cross, this will be the condition, right? 50% will come tall and 50% will appear recessive. Again, the ratio is going to be 1:1. Here the ratio is going to be 1. So he saw that when 75% of the offsprings were made to perform test cross, out of 75%, 25% offspring result was all tall offsprings, and remaining 50% were giving the result 1:1 in the ratio. That means 25 offsprings which are producing all tall offsprings are 25, and 50 are the ones which are giving 1:1 ratio. So this means that Now see, out of 100 offsprings in F2 generation, check this, 25 though were clear having this genotype, 50 were having this genotype. How? Because the result of their test cross, the result of their test cross was 1:1, where 1 means tall and 1 means dwarf. Correct? Whereas 25 offsprings will have homozygous dominant. How we came to know this? Because the result of their test cross was only 1, that is all tall. Thus you can write the genotypic ratio for F2 generation will come 1:2:1. How will you write? This is the genotypic ratio for F2 generation in a monohybrid cross, which was decoded with the help of test cross. Test cross simply means crossing the unknown offspring genotype with the homozygous recessive parental type. So this is how Mendel came to know that what is the genotypic ratio. Now this is for F2 generation. Now let's check out for F1 generation. Now genotypic ratio of F1 generation will be calculated how? Again test cross. Test cross means what? The organism whose genotype has to be calculated. Unknown genotype offspring into homozygous recessive parental type. Now what will happen beta? If all offsprings of this result, suppose if all offsprings comes tall, then this genotype is homozygous dominant. But if the ratio become 1:1, that means it is heterozygous dominant. So what happened for F1 generation? When genotype was calculated with the help of test cross, the ratio came out as 1:1. Why? Because 50% were coming tall and 50% were coming dwarf. That means that the unknown genotype is heterozygous dominant for the given trait. And you know this, that yes, the offspring of F1 generation was having heterozygous dominant condition for the TG. But the phenotype will be tall. Understood? So what is the genotypic phenotypic ratio of F1 generation and F2 generation? Clear with respect to monohybrid cross? Yes. So this is the monohybrid cross performed by Mendel. Now on this basis beta, on the basis of monohybrid cross, Mendel gave two laws. One, law of segregation. Second, law of dominance. If I talk about law of segregation, then this is a universal law without any exception. So this was the biggest success, making law of segregation, framing law of segregation was one of the biggest success of Mendel. So first I'll be teaching you the universal law, which is law of segregation, also known as law of purity of gamete. Universal means no exception to this law. Everywhere law of segregation is applicable. This was the biggest success of Mendel. Now what law of segregation says? Beta, according to this law, now we know that when an organism is diploid, in a diploid organism, every gene is represented by a pair of allele. Correct? In a diploid organism, every gene is represented by a pair of allele. However, when meiosis takes place, then Mendel said both the alleles segregate, separate from each other without blending, without influencing. Both the alleles will separate out from each other as if they were never together. And each gamete will receive one allele out of the two pure allele. Right? So this is known as law of purity of gamete, because after reduction division, after meiosis, in a diploid organism, when gametes are obtained, each gamete are going to receive one allele out of the two and pure allele, no mixture. And this is universal, accepted everywhere. So what is law of segregation? According to this law, alleles of the gene segregate independently without blending with each other during the time of meiosis. That means let's write it in the form of a diagram also. Like for example, an individual is diploid. Diploid means having pair of homologous chromosome. And on each pair of homologous chromosome, for every gene, there are two alleles. Like for example, so I have taken 3 genes. That is R gene, T gene and Y gene. Suppose all the 3 genes over here are present at 3 different loci. This is locus 1, this is locus 2, this is locus 3. Right? Now, and each gene is present in heterozygous state. Now, beta, when reduction division, when meiosis will take place, so gametes will be formed. Correct? Now, what will happen? Each gamete is going to receive only one allele out of the 2. So yes, what will happen? See, this is the condition. So each allele is going to separate out independently. These are the alleles which are going to separate out independently without influencing each other. Alleles. Now these alleles will now form gametes. What will be the gametes formed? How many gametes? 2 की power 3, because there are 3 genes all present in heterozygous state. So total 8 gametes will be formed. I hope now you can form the gametes. This is homework to form the gametes for the respective alleles. 8 gametes you are going to make. Now I'll even make it why to give you as a homework. So what is the condition? First T gene, then Y gene, then R gene. This is only how we make the gametes. See. So can you see that each allele of the gene has segregated independently? They are not influencing each other. Capital T is not influencing small t. Capital R is not influencing small r. Capital Y is not influencing small y, even when they were together. Correct? Now how the gametes will be formed? See. This is gamete number 1. Correct? Then second gamete. This is gamete number 2. I hope you are able to understand. Then this is gamete number 3, capital T, small y, capital R. Then this is gamete number 4. Now moving towards gamete number 5. This is type of gamete number 5, then gamete number 6, then see gamete number 7, and last is gamete number 8. So these are the 8 types of gamete formed with the help of 3 heterozygous genes present in a diploid organism which segregated independently without influencing each other. Right? Yes or no? Now write one more point. Note point. Each gamete receives only one allele out of the 2. Right? So this is law of segregation, 100% absolutely correct law. Now, then on the basis of monohybrid cross, Mendel also framed one more law by the name law of dominance. Now, this is not a good law, I will say, because Mendel made so many errors. The points which he explained, the postulates which he explained for law of dominance are having so many exceptions. So this law is not that good. Mendel made many mistakes in making this law, which were proved later on. So first we will see what Mendel said, and then we will see why Mendel was wrong in framing law of dominance after studying monohybrid cross. Correct? Now, after studying monohybrid cross, Mendel realized certain points for gene. He said one thing. First of all, according to him, every gene is controlling only one character at a time. So Mendel said, according to this law, every gene is controlling one character and each gene is represented by only 2 alleles, be it in an organism or in a population. Now, according to law of dominance, I am repeating once again, Mendel said that each gene controlling one character is represented by 2 alleles only. Now, if in any diploid organism, if the gene is present in its heterozygous state, then one allele has to be dominant, another has to be recessive. Correct? The dominant allele is going to suppress the recessive allele and the phenotype is going to be of a dominant allele. According to Mendel, Mendel explained qualitative inheritance. He said that even if one allele is dominant out of the 2, then one dominant allele is capable of bringing dominant phenotype. He believed in quality. He did not believe in quantity. He said even if one dominant allele is there, dominant phenotype. Correct? So what Mendel believed? He believed in qualitative inheritance. That is, one dominant allele is sufficient to produce dominant phenotype. Correct? Now, what is this qualitative inheritance? I'll tell you more. So, what is first of all law of dominance? First point what Mendel said, according to this law, each gene is represented by only 2 alleles, out of which one has to be dominant, one has to be recessive. Now, there are 3 types of combination present, either homozygous dominant or homozygous recessive or heterozygous dominant. If condition is heterozygous dominant, as we saw in case of F1 generation, there one allele was dominant, another was recessive. So all offsprings were appearing to be tall. So Mendel said that one dominant allele is suppressing the effect of the recessive allele and all offsprings are coming tall, that means one dominant allele is sufficient enough to produce the dominant phenotype. Hence, he explained the qualitative pattern of inheritance. Correct? Now, because he was studying such 7 characters which were controlled by different genes, 7 genes, 7 character. So, Mendel said, okay, each gene is controlling only one character. Right? Then he said, beta, that every gene is having only 2 allele because the 7 character considered by Mendel were controlled by 7 different genes and each gene was represented by only 2 allele, even in a population. So on that basis, Mendel framed his law of dominance. Now let's see the exception. Talking to the first point, we know the concept of multiple allelism. We know that one gene can be represented by more than 2 allele. It's just that Mendel could not see because the characters, the genes considered by Mendel did not show multiple allelism. So, because of which Mendel did not know about concept of multiple allelism. So, first exception is over here. First exception is multiple allelism to law of dominance. How? Because one gene can be represented by more than 2 alleles in a population. You know that, blood group. Second, he said that each gene is controlling one character. Now, this was not the problem of Mendel because the characters which he chose were controlled by one gene only. But we know that one gene can also control more than one character, pleiotropy. However, Mendel did not observe pleiotropy, so he did not mention about it. But we know we have an exception. Exception. Pleiotropy, where one gene is controlling more than one character. Mendel said one gene, one character, but it is not so. One gene can control more than one character. Exception, pleiotropy. 2 exception. Third, incomplete dominance. Mendel explained qualitative inheritance beta. Mendel said that if one dominant allele is present out of the 2, 100% dominant phenotype. But it is not so. Sometimes one dominant allele is not sufficient enough to bring the dominant phenotype. So, third exception is incomplete dominance. Right? Fourth, in this only, we have one more, that is codominance. Codominance says beta, now Mendel said that if heterozygous situation is there, in a heterozygous situation, one has to be dominant, one has to be recessive. So Mendel said if a gene is present in heterozygous condition, one is dominant, another is recessive. But codominance, here is the exception. Codominance, why? Because if heterozygous condition is there, it is not always guaranteed that one will be dominant, one can be recessive. Both can be dominant or both can be recessive, right? According to Mendel, if heterozygous condition is prevailing, then one allele has to be dominant, one allele has to be recessive. However, it is not guaranteed. AB blood group, both the alleles are dominant. A is also dominant, B is also dominant. So see, heterozygous condition is there, but still both the alleles are dominant. So again, exception. So now you see beta, Mendel law of dominance is full of exceptions. Each line has an exception. First line, multiple allelism. Second line, codominance. Third line, pleiotropy. Fourth line, incomplete dominance. So every line, every point in law of dominance by Mendel has exception. Now, will we say that Mendel did not understand properly? No, it was not the problem of Mendel. Actually, he performed monohybrid cross. And on the basis of that cross, he simply framed the laws. Now, it was in his luck that the characters which he chose did not show these exceptions. Mendel chose 7 characters of P which neither showed incomplete dominance, which neither showed pleiotropy, which neither showed codominance, which neither showed multiple allelism. That is why Mendel did not come across all these points. Hence, he framed the law of dominance. But when later more scientists came, they experimented on different organisms, then they saw that, Arey Baba, this law of dominance is not correct. It is only applicable to those 7 characters chosen by Mendel on P. But as you change the organism, as you change the character, law of dominance is complete failure. There are so many exceptions. Correct? Chalo. Now we will see all these exceptions one by one in detail. First, starting with multiple allelism. Little bit related to multiple allelism I have already explained you. But here I'll help you with the help of an example. First, multiple allelism means that a gene having more than 2 alleles in a population, like for example beta ABO blood group system of human, right? Our blood group system which is controlled by gene I. It is represented by 3 allele in a population, IA, IB and recessive I. Correct? Which can also be written as IO. Understood? So how many alleles are there? 3 allele. So for I gene, the number of allele is 3 in a population. So it becomes an exception to Mendel, that Mendel said 1 gene, 2 allele. But here you see 1 gene more than 2 allele, exception multiple allelism. Understood? Yes or no? Now on this multiple allelism, this blood group system, I want to give more explanation. So for this, we will make the whole blood group chart. We will see that what are the combinations, genotype possible, what are the phenotype possible for this I blood group showing multiple allelism. Now beta, if any gene is showing multiple allelism, then there is a formula to calculate the number of genotype possible for it. So here, genes, to calculate the possible genotypes for a gene showing multiple allelism, for gene showing multiple allelism, we have a simple formula. The formula says n by 2 bracket mein n plus 1, where n is the number of alleles. So here for blood group, what is the value of n? 3. So 3 by 2 bracket 3 plus 1. So 3 by 2 into 4. How many genotypes are possible? 6 genotypes are possible for human blood group system. Now let's check these. So for this, I'll be making a chart first. Now let's draw a chart for complete blood group system. I'll show you what are the possible genotypes and phenotypes. Okay. So beta, there are 4 phenotypes possible. Genotypes is 6, and for blood group 4 phenotypes are possible. You know, A blood group, B blood group, O blood group and AB blood group. Correct? Yes or no? So here I am going to write the blood group Correct? Then what is the antigen present in that blood group? With respect to antigen, beta, blood also forms antibodies. And then we will write the possible genotypes for the specific blood group. And yes, phenotype. So it's clear only. So how many blood groups are possible? 4. A, B, AB, O. A blood group means antigen is A. Antibody is opposite to the blood group B. B blood group, antigen B, but antibody A. For AB blood group, beta, both the antigens are produced, but no antibodies. For blood group O, which is universal donor, no antigen, but both the antibodies are produced. So this is something which you have to remember. Now, talking about the possible genotypes. For A blood group, there are 2 genotypes possible. One homozygous dominant, another heterozygous dominant. So a person having this genotype will produce A phenotype, and person with this genotype will also give A blood group. For B, again, homozygous dominant or heterozygous dominant will give B blood group. For AB, there is only one condition. When both the alleles are dominant, one with IA, one with IB. Here you see the codominance. Blood group will be AB. For O blood group, the only condition is when both the alleles of the I gene are present in recessive state, then the blood group comes O. Now beta, you see A blood group, B blood group and O blood group. All the 3 are following law of dominance. How? Correct. See, law of dominance says that a gene must be represented by 2 alleles. So here you see 2 alleles are there. They can be either same or they can be either different. If the alleles of a gene are different and if they are present in heterozygous state, then one will be dominant, one will be recessive. So check this one. Here, one is dominant allele, another is recessive allele, and the phenotype is decided by the dominant allele. So IA is bringing the phenotype as A, IB is bringing the phenotype as B. Correct? Here also you can see beta in O blood group. Now homozygous recessive condition is there. None of the dominant allele. So no dominant expression and blood group is becoming O. So A blood group, B blood group, O blood group. They are following the law of dominance. But when we talk about AB blood group, AB blood group, they are not following law of dominance. Rather, they are showing codominance. How? Because here you see heterozygous condition is there. Both the alleles of I are different. IA, IB. Heterozygous condition. Now Mendel said if a gene is present in heterozygous state, then one allele has to be dominant over other and expression and phenotype will be because of that dominant allele. But here what happens beta? Heterozygous condition is there. However, both the alleles are dominant. IA is also a dominant allele. IB is also a dominant allele. Therefore, IA is also going to express itself and IB is also going to express itself. Thus, the blood group will be neither A nor B. It is going to be their combination, which is AB. Codominance means when both the alleles in a heterozygous state are expressing themselves equally. None of the allele is suppressing the other one. IA cannot suppress IB. IB cannot suppress IA. Both the alleles will function. They will produce their antigen. So you see, both antigens are present. As a result, antibodies are not synthesized in such blood group. So this is known as codominance, which is an exception to Mendel. First of all, blood group itself is an exception overall as it is showing multiple allelism. Now, in blood group, AB blood group type is more exception as it is also showing codominance. ABO, following law of dominance. This is following law of dominance. But AB is not following due to codominance. Understood? Yes or no? Okay, very good. So now we are going to proceed with the next exception. I hope multiple allelism is also clear and now codominance is also clear. I'll write it over here. Codominance, AB blood group in human shows codominance as when, let me write it over here, as when gene I is present in its heterozygous state, comma, both the alleles are equally expressing. Both the alleles are equally expressing, bracket, none of them is getting suppressed. None of them is getting suppressed. So this is known as codominance. Like I'll explain you in more detail. See, IA allele, capital IA allele beta expresses itself, produces proteins. Those proteins produces an enzyme called N-acetyl galactosamine. N-acetyl galactosamine transferase enzyme. Right? This enzyme beta, if this enzyme is produced in any organism, then he or she is going to produce A antigen in the body. Correct? That means which blood group? A blood group. Now, if IB is present, then which enzyme will be produced? N-galactosyl transferase. Or remove N, only write galactosyl transferase. This enzyme beta is responsible for the production of B antigen, making the blood group B. Correct? Now organism having both IA, IB will produce both the enzyme, thus both the antigen, thus blood group will become AB. Now organism having small I, no enzyme, no antigen, O blood group. Clear? So IA allele means production of enzyme N-acetyl galactosamine transferase. IB allele means production of enzyme galactosyl transferase. These enzymes are respectively responsible for producing A and B antigen. If both the alleles are present, both the antigen, blood group AB. If one of the allele is present, then accordingly antigen is produced and accordingly blood group is decided. If both the alleles IA, IB not present, small I present, no enzyme, no antigen, blood group O. I hope you have understood the first two exceptions of Mendel, that is codominance and multiple allelism. If yes, then we will move towards the next exception of Mendel, that is incomplete dominance. Now Mendel said beta that if one dominant allele is present, then it is sufficient enough to produce the dominant phenotype. Correct? He said na, if out of the two allele, one is dominant, then surely organism is going to bring the dominant phenotype. Correct? However, it is not true. Mendel explained qualitative inheritance pattern. But there was a scientist by the name Correns, Carl Correns, while he was studying on the flower color of snapdragon, he observed that one of the dominant allele is not sufficient enough to bring the dominant phenotype. So that is known as incomplete dominance, which is actually a quantitative inheritance, because the quantity of dominant alleles will decide the range of phenotype, as the number of dominant alleles will decide the type of phenotype, because one dominant allele is not sufficient enough to bring the dominant phenotype, is what it was explained by Carl Correns while working on the flower color of snapdragon. So he started to make the cross. First, same pattern. This fellow Carl Correns also made the monohybrid cross following the instructions given by Mendel. So what is the first step in monohybrid cross? Selection of the parent, and parents have to be always pure line. Okay, so now Correns also made the monohybrid cross following the instructions of Mendel by using which gene? By using R gene, that is controlling the flower color of snapdragon. So what is the first step of making monohybrid cross? First step is selection of parent, and parents have to be always pure line. So homozygous dominant male crossed with homozygous dominant female. Correct? If this is the condition, then phenotype was red colored flowers. If this is the condition, then phenotype is white colored flowers. So parent selected, genotype clear, phenotype clear. Now what will happen? The second step is performing cross pollination. Correct? So gametes are obtained here. Gamete here, this is the gamete. Now gametes will be made to fertilize to obtain F1 generation. This will be the genotype of F1 generation. What is the genotype of F1 generation? What Correns expected? Correns expected that all offspring should come red in color. Yes or no? This was his expectation. However, none of them were red nor white. Some intermediate color came, that is pink. So Correns was expecting that all offspring should come red colored. Why? Because Mendel said when pure line parents are crossed, then all offspring brings dominant phenotype. Right? So here also Correns was expecting, because he is following the same steps of Mendel. Pure line parent are selected, red colored flower crossed with white colored flower, homozygous dominant, homozygous recessive. So he expected that all flowers should come red. But it did not happen. Actually, some intermediate color came. All offspring seem to become pink in color. So this is actually intermediate color appeared. Here, Correns got shocked that, oh, Mendel made a mistake or he made a mistake. So to ensure that, he continued the experiment of monohybrid cross. Then what is the next step? Selfing. Okay. Selfing of F1 generation. So what will happen now? Two types of gametes. Same two types of gametes. Punnett square. Correct? Yes or no? Heterozygous condition for the gene are. Now what he was expecting? That F2 generation phenotypic ratio should be 3:1. That is 75% should bring dominant phenotype, which is red color, and 25% should bring recessive phenotype, which is white color. But he was shocked to see that F2 generation phenotypic ratio changed from 3:1 to 1:2:1. That means 25% came red, 50% came pink, and 25% came white. So the phenotypic ratio changed from 3:1 to 1:2:1. You can see over here very clearly. These are red, these are pink, and these are white. Now he could only see the phenotype. So to confirm the genotype, he performed the test cross, and in that he came to know that the genotypic ratio is also same. 1:2:1. So beta, in case of incomplete dominance, the phenotypic and genotypic ratio for F2 generation came to be same. The result is in front of you. He also did the same test cross to calculate the genotype of each individual, and he saw that the result is same, 1:2:1. The only difference was in phenotypic ratio. Phenotypic ratio instead of 3:1 came as 1:2:1. So Correns got one thing, that Mendel qualitative inheritance is wrong. Always one dominant allele is not sufficient enough to produce the dominant phenotype. As here, what happened? What happened in case of flower color of Snapdragon? One dominant allele. See, when the heterozygous condition was this. So one dominant allele did not or is not sufficient enough to produce red color flower. That means to produce red color flower, both the alleles should be dominant. Both the alleles should be dominant in order to produce red color flower. If one allele is dominant, then neither red nor white, some intermediate color will come, which is pink. And if both are recessive, then in that case white color flowers will come. So that means to produce dominant phenotype, one dominant allele is not sufficient enough. I hope you have understood incomplete dominance, which is one of the exception to Mendel's qualitative inheritance. as it explains the pattern of quantitative inheritance where number of dominant alleles decides the type of phenotype. Now, I just hope you have understood the first three exceptions by Mendel, the first three exceptions to Mendel's law of dominance, that is incomplete dominance, multiple allelism, and codominance. Talking about the last exception to Mendel theory is pleiotropy. Pleiotropy, see, Mendel in his law of dominance mentioned one point that every gene controls one character only. Like for example, T gene is controlling height, Y gene is controlling color of the flower in case of P. So according to Mendel, one gene at a time can control one character only. But we know this is not true. Actually, one gene can control more than one character. So again exception come, and this exception is known as pleiotropy. Almost every gene is controlling more than one character. So majority of the genes in our body, they are showing pleiotropy. One example which I am going to discuss is taken from the pea plant itself. Had Mendel chosen seed size instead of seed shape, instead of seed color, then Mendel would have also seen pleiotropy as well as incomplete dominance. But since Mendel chose two seed color as seed shape and seed color, he did not took seed size. So Mendel got saved from pleiotropy and incomplete dominance because seed size and seed shape in P shows pleiotropy. These are two different characters but controlled by the same gene B. Correct? So example, seed size and seed shape both are controlled by same gene in pea plant. So Mendel was lucky enough to not select seed size as one of the character, rather he chose seed color. So he got saved from pleiotropy as well as incomplete dominance. I'll tell you how. Now, seed size because also shows incomplete dominance. Now, Mendel did not choose it. So he did not got an opportunity to see incomplete dominance and pleiotropy. Okay. Now, both seed size, beta, and seed shape, they're controlled by same gene B. B gene beta in case of pea plant is responsible for the production of proteins, which in return forms enzyme called starch enzyme, starch branching enzyme. So B gene produces proteins, which in return produces enzyme SBE, starch branching enzyme. Right? Now, this enzyme is responsible for controlling both seed shape and seed size. How? First, we are going to discuss about seed shape. Now, seed shape is normal, showing law of dominance. There are three conditions, either homozygous dominant, heterozygous dominant, or homozygous recessive. You know, seed shape is one of the character taken by Mendel. So it is going to show law of dominance. It is going to follow the law of dominance, which says that if one of the allele is dominant, dominant phenotype is going to come. So here the shape will be round. Normally it is homozygous dominant. Here homozygous recessive, the shape will be wrinkled. And according to law of dominance, if even one of the allele is dominant, then surely phenotype is going to be of dominant nature. So here also it is going to be round. So seed shape showed exactly the same thing as Mendel suggested. However, when we talk about seed size, then seed size beta shows quantitative inheritance. First of all, there are two different characters, both controlled by B gene. So they are showing pleiotropy. Now in this also, seed size is showing incomplete dominance. How? Now there are three genotype possible: homozygous dominant, heterozygous dominant and homozygous recessive. If Mendel is absolutely correct, then pure line homozygous dominant seeds will be large sized, which is actually homozygous recessive will be small sized. However, if Mendel was absolutely correct and there was no quantitative inheritance, there was no incomplete dominance, then even here, where one of the allele is dominant, size of the seed should have been large. But here, the size is neither large nor small. Why not large? Because two dominant alleles are not present. Why not small? Because at least one dominant allele is present. So size will be intermediate. So the seeds which are having heterozygous arrangement of B gene, that is capital B and small b, their size are neither going to be large nor small. They will show a intermediate size. Correct? This shows that number of dominant alleles are influencing the nature of phenotype, because one dominant allele produces specific amount of protein, producing specific amount of enzyme, and that specific amount of enzyme is not capable of producing large seed. When two dominant alleles were present of B, then maximum protein, maximum enzyme, maximum size. One dominant allele, medium protein, medium amount of enzyme, medium size. No dominant allele, homozygous recessive condition, no protein, no enzyme, size is small. So the size is under the control of the enzyme. Starch branching enzyme SBE controls the size of the seed. Maximum amount of enzyme, maximum size. Intermediate amount of enzyme, intermediate size. No enzyme, small size. Clear? And the production of enzyme depends upon the number of dominant allele. Two dominant allele, maximum synthesis. One dominant allele, intermediate synthesis. No dominant allele, no synthesis, small size. Clear? So seed size is also showing pleiotropy and is also an example of incomplete dominance. So can't you say that Mendel was lucky enough to not choose seed size? If he would have chosen seed size instead of seed color, he would have also seen pleiotropy and he would have also seen incomplete dominance. And he could have not proposed law of dominance having so many exceptions. Understood all the exceptions to Mendel theory? If yes, then law of dominance is done along with the exception. Mendel's monohybrid cross is also completed. For today, we are going to keep it till here only. Correct? Introduction to dihybrid cross, I am not going to give you today. This portion, complete dihybrid cross, we will do from our next lecture. So what we have completed today, beta? Today, we have understood what is monohybrid cross. Then test cross we have seen to calculate the genotypic ratio of or genotype of unknown organisms. Then you have seen the laws proposed by Mendel on the basis of monohybrid cross, that is your law of segregation, which is 100% correct. Then law of dominance. In law of dominance, you have seen all the exceptions which are prevailing to Mendel's this law, that is incomplete dominance, codominance, multiple allelism and pleiotropy. I hope you all have understood so far. Please read it once from your NCERT to make sure that you have understood it well. And to solve, I am giving you some questions as homework, and remaining you can also solve more questions. So these are the homeworks for you. I hope you will do them And with this, we come to the end of our today's lecture. Hope you all understood it well. Thank you for joining me. See you in the next class. Bye bye everyone. Take care.